Hi, everybody. Welcome to my daily therapy session slash virology ranting session. I'm Dr. Angie Rasmussen, and I'm really glad that you joined me today because there's an incredibly important topic to talk about. But first, I'm going to get a couple other items of business out of the way. We have a still ongoing Ebola situation. The U.S. response to this has not gotten any better, although now I guess we are going to appoint some Ebola Czar. Unclear if this is going to be penis enlargement expert and urologist Admiral Brian Christie, who previously managed the hantavirus situation at the biocontainment unit in Nebraska. There hasn't been any onward hantavirus transmission, not that we expected there to be any. But, he's done such a bang up job at that who knows, maybe they'll appoint him to the position of Ebola Czar. Maybe they'll appoint somebody else like Casey Means, who, got knocked out of being Surgeon General because she's a metabolic wellness grifter and a total quack. That's what's going on with the Ebola outbreak. There's also something really important that I'm going to continue talking about to put on your radar, which is the fact that Russ Vought, the Office of Management and Budget Director, is trying to make very vast and sweeping changes to the way that federal grants are issued. This is not just research grants from NIH. These are federal grants to do things like build roads, maintain schools, maintain basic infrastructure that we all enjoy having in our lives. I think that's actually quite bad. We have until July 13th to comment on this. So I will be dropping a link into the Substack post where you can go ahead and do that on the Federal Register, as well as linking you to some advice on preparing this comment and what you should kind of talk about. But today, we are going to talk about something that I think is a really hugely important issue. And this would be a huge issue to me, even if it didn't affect people that I happen to know and have worked with for quite a long time. Basically, I found out yesterday, thanks to a press release, as well as some gleeful tweets from Laura Loomer, that my colleague and friend and longtime collaborator, Vincent Munster, a virologist at the National Institutes of Health, National Institute of Allergy and Infectious Diseases facility, Rocky Mountain Labs in Hamilton, Montana, was charged in federal court in the Eastern District Court of Michigan with attempting to smuggle Mpox virus into the U.S. And lying to Customs and Border Protection officials about it. Now, he was actually only charged with making false statements to CBP, but their own complaint really demonstrably shows that this is in fact not true and that these charges are manufactured. And I'm going to talk a little bit later on about why I think that is. But I think everybody really needs to be incredibly alarmed about this, because this is the first time, to my knowledge, that one of my colleagues has been charged with a federal crime when very clearly no crime has actually been committed. And they are actually filing charges that are completely fabricated and that their own affidavit demonstrates are fabricated. So let's get into all of that. Here's what happened. My colleague, Vincent Munster, and his postdoc, a postdoctoral fellow working in his lab at NIAID, RML, went to the Republic of Congo for like a couple weeks. There is an outbreak of clade 1B Mpox virus spreading in the Republic of Congo, as well as the Democratic Republic of Congo. There has been a huge problem with emergent Mpox outbreak. The whole situation has been incredibly complicated. It has spread to other countries. It's spread around the world. Mpox is a terrible disease. It's usually not fatal, but it can be. Responding to an outbreak, again, in a country that has historically been heavily dependent on U.S. Foreign aid and support for their health systems, their infrastructure, and their capabilities of investigating and containing outbreaks, It actually makes sense. And in the before times, my colleague Vincent used to do exactly this. He would go to countries where outbreaks were occurring because he is an expert on emerging viruses, and they emerge. He goes to those places, he gets samples, and he also has assisted extensively with diagnostic testing. During the West African Ebola epidemic, Vincent went, along with many of his other colleagues at RML and NIAID, went to West Africa and did a lot of diagnostic PCR. They also helped train people to do diagnostic PCR, so therefore they could increase the countries that they were working in's capacity. And that appears to be what they were doing in Republic of Congo with regard to this Mpox outbreak. So they were pulled aside at the airport in Detroit when they re-entered the country. And here is what the documents say about that. This is from the affidavit of the FBI agent who investigated this case. They said that basically Vincent and his postdoc stated that they only had diagnostics. They were not transporting so-called biological samples, which would be like if they went there and got samples and were bringing them back to the US to do more testing on them. That requires different kinds of permits. And it also, you need to be able to show that you are inactivating the samples properly because clade 1 Mpox is what's called a select agent in the US. So there's a federal law called the Select Agent Law that governs how we handle certain pathogens. Now, I had select agent clearance from 2016 to 2021. Since 2021, I've had a security clearance under the Canadian equivalent of select agent law. We have an equivalent law in Canada that's called the Human Pathogens and Toxins Act. So I actually know quite a lot about select agent law because, in fact, through my collaborations with Vincent and some of my other colleagues at RML, over the years, I have received a number of inactivated samples of Ebola virus, not of Mpox, but Ebola virus, MERS coronavirus, other viruses that I have worked with in collaboration with them. And the reason that they are able to send these samples to me and I don't have a maximum containment lab at my disposal is because they've been inactivated using a validated procedure. And furthermore, when select agents are inactivated, unless you can use their genetic material to reconstruct the pathogen using reverse genetics, they are no longer regulated as a select agent. So that's really, really important to understand for this, because while Mpox clade 1 is a select agent, these samples were inactivated. And this actually says that explicitly, that these samples were in fact deactivated according to the Department of Justice's press release, as well as this complaint or these charges. So the bottom line is that Mpox is a DNA virus, and no matter what the sort of AI biosecurity community tells you, you cannot actually just reconstitute Mpox with Mpox DNA. So Mpox clade 1b infectious virus is in fact a select agent. When it's deactivated, because you cannot use that DNA to directly reconstitute infectious Mpox virus. So these were not samples that were going to be regulated or that needed to be regulated as select agents, as long as they were inactivated following a validated protocol. So it's hard to say what exactly these samples were, because the documents that are mentioned in the complaint include a sample inventory. And it seems that at one point these samples were grown in cell culture. They are samples that contain Mpox DNA that have been inactivated using what I recognize to be RML standard procedure for inactivation. Now, I think I have actually, let's see, oops, I did not. I accidentally didn't upload that file, so we'll just talk about it. But basically, these samples underwent a procedure where in a biosafety cabinet at RML, they extracted the DNA using a buffer called APL buffer. That's made by a company called Kaigen, and they add ethanol to it. And this is a standard procedure for inactivating samples. Now, it's not just enough to actually use this procedure to inactivate samples, and this differs from facility to facility and country to country, how this is done. But in both the US and Canada, the way that it has been done over the last 10 years or so is that you do the inactivation procedure, you develop the procedure, and then you test it over and over again in the containment laboratory. So you actually make sure that the inactivation procedure works and works reproducibly, works no matter what, with the actual pathogen that you're trying to inactivate. So that way you can prove that every single time you follow this procedure, that pathogen is going to be completely inactivated. And that's how we make sure that when we are sending samples of Ebola or Mpox or something that's really pathogenic to somebody and we're saying this is an activated sample, you don't have to handle this in a containment lab. And it's also not a risk because somebody with nefarious intent could not get their hands on it and use it to actually generate the infectious pathogen from it. We know and we can prove that this procedure does that every single time. And so every facility will do this over and over again. We don't like look at somebody else's protocol and say, oh, we can just do that here. It'll probably work. No, we have to test it ourselves extensively. This is actually something that takes up a lot of time in your lab if you do work in a containment laboratory. And this is something that my lab has, in fact, for Mpox, using a different inactivating buffer, we've spent quite a lot of time and generated quite a lot of data on. So these samples had been inactivated using this validated procedure at RML. Now, again, as I said, they were originally cells that were infected with Mpox as best as I can tell. And so what happened then was Vincent and his postdoc were coming into the country. They had these samples with them, and they said to CBP when they were questioned about it, that these samples were diagnostic samples. So they were diagnostic tests. And the FBI is contending that they lied about that because they were actually, pathogen samples, biological samples, meaning they were going to be used for research, and they were trying to illegally smuggle them in. And at one point in the affidavit, Vincent actually says, when I bring samples back, because a big part of Vincent's research is studying different animals, different hosts. This is why we collaborate. Our research programs are very similar and complementary. We focus on similar topics. But he goes out and finds bats, other species of hosts, and of course people too, and collects them and tests them and looks for emerging viruses, tries to understand better how these viruses might emerge naturally. This is a perfectly legitimate field of study. It's a really important field of study. And I'm not saying that just because that's my field of study too. I'm saying that because Mother Nature is full of all kinds of horrible emerging viruses that could really, really screw with global health, with the entire world. We already saw what happened during the COVID pandemic. We're seeing what's happening right now in this emerging Bundibugyo Ebola virus outbreak that's occurring in DRC. And we've in fact seen this in the Mpox outbreak that has occurred in DRC and Republic of Congo and throughout different parts of Central Africa. There's separately Mpox outbreaks occurring in West Africa too, of the clade 2 the clade 2 version of this virus, different evolutionary branch. We're seeing, the point here is that we're seeing these emerging viruses popping up more and more often, more and more frequently, and understanding where these viruses are in nature and how we might be exposed to them is really, really critical for preventing a new emerging virus from popping out into the human population and really screwing us all up. So there's nothing sinister about what Vincent was doing, and it actually He was bringing diagnostic testing kits to a country that was being ravaged by an mpox outbreak and really needed those diagnostic testing kits. And one thing we can see from the current Ebola outbreak that's happening is that if you don't have accurate and rapid molecular diagnostics, you are in real trouble because you can't identify actual cases. And with mpox, that is a huge problem because while people do eventually usually develop this rash, they don't always, and there can be a really, really long incubation period during which people can actually transmit mpox. So unlike Ebola, it's not just when you become symptomatic with mpox, you can actually be contagious for a very long period of time. So having accurate molecular diagnostics readily available in a place where an outbreak is occurring is really important for the response and really helpful for the response. The way that this has been characterized in this complaint is that Vincent lied about bringing these diagnostic tests back, and they tested it, and there was mpox DNA in there, and they were deactivated samples, but somehow that means that because clade 1 mpox is a select agent, that Vincent was somehow trying to sneak it in rather than send it with World Courier like he normally would do with his actual research samples. First of all, there's no reason why you would need to do that. Second of all, Vincent has been doing this for a very long time, probably for the last 20 years. I'm not sure exactly when he started working at RML, but he's been doing this type of work for a really long time. He's done it with MERS, Ebola, mpox, you name it. He's been doing this very safely, and he's been contributing importantly, really, really good research that has advanced our field. During the West African Ebola epidemic, he not only did great Ebola research, he also assisted with diagnostic testing. He assisted again with diagnostic testing, with efforts to test the vaccine in the Democratic Republic of Congo in 2018. He has done a lot of great work in terms of responding to these outbreaks. And that's what he was doing in Republic of Congo. So when he told those customs officers that these kits contain diagnostics and yet they contained this mpox DNA, what could that possibly be? Well, guess what? When you do a diagnostic test, you need to have these things called controls. And controls are things that what the test result is going to be. One of the easiest ways to make controls for a new diagnostic test that you're developing, a PCR test, for example, if you are a virologist, is to grow up some of the virus that you're testing for, isolate its genetic material, and use that as a control because you already know it's the virus that you're looking for. You already know that it's going to be positive. And when you grow it in cell culture in a lab and then isolate it, you know for a fact that you are going to have a lot of that material in there that will test positive. Similarly, you do that with a different virus and you use that as a negative control, which is equally as important because you want to make sure that your test isn't going to pop false positives for another DNA virus, for example, an alpha herpes virus like chickenpox, which this complaint wrongly implies is also an orthopox virus. Chickenpox is the name of the disease that varicella zoster virus causes. Varicella zoster virus is a herpes virus. It is a DNA virus, but it is not, importantly, an orthopox virus, which is what mpox is. Mpox and herpes viruses are completely different, other than both of them have genomes that are made out of DNA. This was a negative control in my view of this, or my assumption. I don't know that for sure. Again, we haven't actually seen the full list of documents and what they were, but reading this and knowing how diagnostic testing works, knowing the type of work that Vincent has done, knowing what inactivation protocols are like at RML, and also knowing that at RML, in the 15 years that I have worked with the scientists there at NIH, they are probably the most fastidious in terms of making sure they have all the paperwork done. They have crossed all the T's, they have dotted all the I's. They are exceptionally focused on making sure that they are doing their work safely and that they are getting maximum value out of taxpayer dollars that support their research for the American people, because indeed they are government scientists and people go into public service for a reason. They go into public service because they want their work to benefit the people, the public, that are supporting it. They know what the mission is, and that is what I have known Vincent Munster to be since we started working together in 2012. So I really want people to understand that when they say that Vincent lied about having biological materials when he said he had diagnostics, all of the documents that they refer to in this complaint or in these charges are consistent with exactly what Vincent said they were. They are samples that are being used for diagnostic testing. There was a whole thing in there about, oh, there's some boxes of microcentrifuge tubes that are labeled funny and that are mixed up. They're not in numerical order, so that's suspicious. No, that's actually not suspicious because there are all sorts of different coding systems. And sometimes if you're working with a lot of samples, you have to randomize them to make sure that you're not seeing what's called a batch effect. So if you run a bunch of samples all at the same time, sometimes you might have an assay specific effect, like the PCR machine is half a degree hotter or something like that time around. You see from one batch to the next of samples that sometimes there can be little fluctuations in the results that you get. The way that you get around this is not only by using controls and also using other types of controls that will control for the technical aspects of the assay, but you also do a thing called randomization. Whereas if you have a bunch of samples, you don't put all the same samples from one group into the same batch. So that way you have samples just chosen randomly. They're ending up in different batches. And then you can look and see by comparing one sample in one group to the next, like a technical replicate, you can see actually what the batch effect is like and you can correct for it. So having boxes of tubes that are labeled with numbers that are mixed up, it seems perfectly consistent with me with having diagnostics and in fact with training people to use a specific diagnostic testing kit. I see absolutely nothing here that suggests that Vincent was trying to smuggle a pathogen into the country, which he wouldn't have to even smuggle into. Vincent works at the National Institutes of Health in one of their maximum containment laboratories, and he actually has access to all of these pathogens already, which is where these biological samples that were actually diagnostic testing controls were generated. So what's the take-home message here? That was a whole long spiel about all of this technical stuff that proves that Vincent didn't commit a crime to begin with, didn't lie, which is the crime that he's being charged with, as well as his postdoctoral fellow, who, is, I think he probably didn't sign up for this when he came to do a postdoc at the NIH. That's for sure. Being targeted by the government because you are politically inconvenient, or in Vincent's case, I think convenient. Because here's what I think is going to happen. I think that these charges are pretextual. Part of this is that Vincent, I think, is going to probably be accused of having some role in starting the COVID pandemic. And that is because, again, Vincent studies emerging coronaviruses, just like I do, and just like many of my other colleagues do. That means that we've looked at SARS coronavirus, we've looked at MERS coronavirus. There is no evidence whatsoever that Vincent or anybody else in the US, in China, anywhere, had access to a progenitor virus to SARS coronavirus 2, which is exactly what you would need to have in order to start the pandemic. There's no evidence that Vincent had anything to do with the emergence of SARS coronavirus 2, but I think that that's what he and other colleagues of mine are going to be accused of. Other colleagues of mine, and possibly me, doubt it, but maybe, are going to be accused of covering it up. And we've already seen people who are in this orbit. My colleague and friend, David Warrens, for example, was correctly charged with trying to evade public records laws. However, should he, as a 78-year-old lifetime retired service member in the public health service, face 58 years in federal prison for just basically wanting to talk shit with other academics and not have it be public? It wasn't right for him to ask to evade, use Gmail instead of his work email so that it wouldn't show up in a public records request. But is it, does that punishment fit the crime? In this case, however, this is even more alarming because Vincent did not commit a crime. This complaint, this entire complaint, the proof that he committed the crime of lying to Customs and Border Protection officers is actually proof that he did not lie. From my technical reading of this, from my expert knowledge at knowing what this type of stuff is and knowing what type of work Vincent did, because I've worked with him for the last 15 years, there is no way that this was anything other than what he said, diagnostics. When RML provided a document that said, what are these samples? It says they're used in diagnostics. These were diagnostic testing controls, which means that Vincent didn't lie and he didn't commit any crime, which means that the government has manufactured this evidence. They are, bringing charges against him, and there's no reason for it. This is the most undemocratic thing I've ever seen, and it's frankly really, really terrifying because Vincent is essentially being set up to achieve a political goal. Now, what I think that political goal is that if the pandemic started with a lab leak and it started with an NIAID employee, let me remind you also of who the director of NIAID used to be. That would be one Anthony S. Fauci, who, Trump and the entire administration despise. So they want to pin the pandemic on Fauci. So what better than a foreign national working at NIAID who does all this scary virus research, studied SARS-related coronaviruses and other emerging coronaviruses before the pandemic, associated with other people who are also being accused of reckless virology, who were funded by NIH and their external academic labs. All of this is going to motivate people to say, and I think this is the idea, oh, we don't want to be doing any more of this virology research. And in fact, all research is bad. All research could lead to accidents. This is really terrible, and we shouldn't allow these reckless people to be doing this work. What is the point of all of this? Where is my new drug or whatever? Why did we have a pandemic? Because all of us suck at our jobs, I guess, and we're all trying to start lab leaks because we just want to do reckless virology all the time. None of that's true. But you know what shutting down research would do? It would return a lot of money back to the executive branch, potentially. If we said we're not having a national research program anymore, Congress has no budget to appropriate for it. And guess where all that money sits? With Office of Management and Budget Director Russell Vought, who wants to completely can all research, who wants to completely can all research grants because they want that money in the executive branch to consolidate and centralize power, undermine our democracy even further, which is exactly what this does. This is arresting a scientist for being a scientist who happens to work for the U.S. government on behalf of the U.S. people. And this should terrify everybody. They are going after you if you just happen to be in the wrong place at the wrong time doing your job, which you have done in an exemplary way for your entire career. So I want to just let my colleague Vincent Munster know that I have so much respect for you, and I'm so sorry that this is happening to you. I am here to speak up on your behalf and on behalf of all of our colleagues who are being unfairly targeted by this administration, and on behalf of all Americans who are being unfairly targeted and set up by this administration in its efforts to further undermine democracy and impose authoritarianism on all of us. So with that, I'm going to sign off because in about 45 minutes, I've got a whole other shit show to deal with because Turbo Cancer Day is in session over at the Homeland Security and Government Accountability Committee at the Senate. Today, the Permanent Investigation Subcommittee is going to be having a nice little chat with a bunch of different anti-vaxxers who are going to make a bunch of bullshit turbo cancer claims. So me and hopefully Jessica Malati Rivera, if she can join me, she's traveling today, will be debunking those all afternoon. So stay tuned for that. You know, if you want to know more about what's going on with these attacks on science and democracy, I'm going to continue talking about it and writing about it. So continue to subscribe and follow for more information. I'm getting better at this. Okay, thanks, guys. See you tomorrow.